Good morning. Good morning. Good morning. Good morning. It's good to see you all again. It's good to be back. And it's amazing to see that in the meantime, you've been keeping your word finding skills tightly tuned and honed. This is impressive. Today, this is kind of the scope of today's lecture. It sounds to me like a band. If Peach Pit can be a band, then Gnathiferans and the Lophophore can also be a band. Right? Wouldn't you go out to see Gnathiferans and the Lophophore? All right. Put your left hand up. That's my right hand. Smith fails. Put your left hand up. Pat yourself on the head for being the winner and still champion at the fastest class at finding words ever. And here we go. I see some of the questions, and thank you for those. I didn't find any more purple socks, but I did sit in my board meeting for the NGO that I work with, the Guanacaste Dry Forest Conservation Fund, with Dr. Jorge Cortez, who you met in that lecture, with, hold on a second, I want to go check out that little purple, that little pink thing over there. I love that moment. It makes the hair on my head stand up. You don't believe that. So it makes the hair on my arms stand up. But it was amazing. It was some field work, some long-term data collection, both with insects, temperature, and precipitation, and how the communities of insects are changing naturally with elevation and being forced through a filter of the climate crisis, and collecting data about precipitation and temperature to convince people of the changes that are happening here in Central America associated with the climate crisis. Lots of insects, lots of ants, lots of amazing things. And being part of this not-for-profit organization, this NGO that pairs with the national park, is one of the honors and privileges and joys of my life. So the more that I can help with that, the better. So you can scan it and find out about the GDCF there later on if you would like. So thanks for asking. Previously on Zoology 2700, we were kind of moving into the bilateria. We were leaving behind the cnidarians and the placozoans and the ctenophores, right? Oh my God, ctenophores! And the introductory taxon that we settled upon with the bilateria were the platyhelminth flatworms. And we focused, as kind of the narrative story, the narrative arc of this course, we focused on the parasitic lineages, the monogeneans, the trematodes, and the cestodes, right? And we saw these amazing scolices and adaptations associated with being a parasite and not being knocked off if you're an external parasite, or not being knocked off if you're an internal parasite, like that cestode. And I didn't get to tell you all that I wanted to about trematodes. So here we are today with this one more story, because it's kind of brilliant. We talked about cnidarians together, right? We talked about the Neuston, the organisms at the top, the interface of the ocean and the air. And we talked about bluebottles, this kind of hydroid colony that lives there. This, to me, and to many people in the world when it was discovered in Okinawa, Japan in the early 2020s, looked like a scyphozoan until you zoomed in. And then it doesn't. This little organism that was found kind of defied identification for a long time because people looked and were like, I don't even know what phylum to put that in. So this enigmatic. And what turns out is that this is not an organism. This is an aggregation of trematodes, of cercaria larvae. Cercaria larvae that are playing different roles, that are being passively carried on the inside and moved by the beating of the tails on the outside. So this is the kind of phrasing that the authors use, sailors and passengers. And they belong to the same species. And this kind of division of labor is the first time that's ever been seen in a trematode. Like, the social nature of this parasite is kind of bananas. Why are they doing it? It seems that it's an effort to attract fish, to attract the next host, to manipulate the behavior of the next host that those cercaria larvae need to embed themselves in to continue their life cycle. Isn't that amazing? That's amazing. So we couldn't leave behind the trematodes without talking about this kind of Okinawa aggregation. So trematodes, WTF. What are we doing today? We're going to talk about Gnathiferans and the Lophophore. The Gnathiferans are two phyla. Well, they're three, right? I do this often. I'll say it's 2 phyla, but we only have time to talk about 2 of the 3. We're going to talk about rotiphores and Chaetognaths. Rotiphores, I'm going to guess most of you have heard something about before. Chaetognaths, I'm going to guess most of you haven't. So I'm going to evangelicalize about the importance, particularly in the plankton, for the predatory Chaetognaths. We'll talk about aggregates, how we now feel that they are aggregated. And then we're going to set the stage for Wednesday, when we start talking about phyla that possess a lophophore, like brachiopods and pheronids and bryozoans. So that's where we're going. My hopes, and so where does this class fit under the big arching over umbrella of the class, is that you can use these 3 active verbs to think about Gnathiferan evolution, to think about causes or the placement of Chaetognaths within the bigger phylogeny of the animals, and consequences for decisions regarding where we place it here or there. And quite analogously, I'm going to think about the phylogenetic placement of the morphological feature, so not the taxon in this case, but the feature of the Lophophore. All right. So we're informing our examination of the morphology with the hypotheses of phylogeny. We've done this before, and we will keep doing it. So our roadmap through the invertebrate, the morass of invertebrate animals, is, of course, the Dunn et al. Phylogeny. Now, when we're talking about Gnathiferans, we're talking about, well, first the derivation. So Greek, a combination of Greek and Latin. So Gnathos, Flanders and Swan, like I'll make a pop culture reference, but from popular culture of probably 90 years ago. I'm a Gnu. How are you? So this is a Gnathos, meaning jaws, and Phara, mean ferrous, to bear. So jaw-bearing things. So we'll talk about this kind of the wheel organ, the really, really interesting wheel organ of the rotifer. And they sit kind of right here. So there's 3 taxa for sure. And then there's a question mark up here by the Chaetognath. And I'm not going to bury the lead. 12 years ago, this was an outstanding question. Where do Chaetognaths fit? Dun, dun, dun. Like the mystery of the tiny arrowworm, as opposed to the mystery of the purple sock. And we're going to solve it today. Or we're going to examine the best contemporary evidence that supports a new placement from where it is right over there. All right? Well, let's go. So this is a couple of different chapters in your Brusca, and this is an examination here of the case, the block that lists out in great detail, and detail that I don't expect you to reproduce, particularly associated with names, some of the synapomorphies that characterize this phylum. So they're a triploblast. They're a bilateral, unsegmented thing. So this is not a worm. They have a complete gut. Remember how important having a gut and having whether it's complete or not, and that it doesn't appear and disappear like those silly ctenophores is. That they have adhesive glands, that they have a syncytial epidermis. We were introduced together to that term syncytial, or we reviewed it last when thinking about the trematode or the platyhelminth last week. They are big enough that we start to see these terms. So like protonephridia. So protonephridia is an excretory organ, but they don't have circulatory or gas exchange organs. So we're starting to see organisms become biggish and complicated-ish enough that we need to see, as we did with the platyhelminth, where diffusion doesn't cut it, and we need to see some of these associated organs and structures to excrete toxic byproducts of metabolism and help get the beneficial food and oxygen to tissues. Males, generally reduced or absence. Parthenogenesis is common, so the production of lineages of new offspring without any need for a male at all. They inhabit marine, freshwater, semi-terrestrial, sessile, or free-swimming, so a whole bunch of diversity of forms and features. And you can see some of those forms and features right there in this beautiful SEM. Here's my kind of breakdown of some of those interesting synapomorphies. They have this ciliary organ, this wheel organ on the head, and it's right there underneath that counting ticker. So rota is Latin for wheel, but the ciliary corona is what sets up feeding currents into a mouth in the center, and we'll take a look at that. They have this retrocerebral gland, that's what that's pointing to there, that secretes mucus. And then they have, I'm sorry, the arrow is actually to the vitellarium, where this syncytial structure in females that produces eggs. And they can reproduce with parthenogenesis. Let's take a look at a cross-section of the body. One of the things we're going to talk about in our time with the rotifers is reproduction, dealing with harsh environments, and movement, and distribution. One of the 5 things. The arrangement of the muscles here is a real clue as to how we're looking. So there is we've got circular muscle and longitudinal muscle. Now, you remember, those are what the phrase I keep using again and again is antagonistic, meaning kind of muscles at right angles. So if you look through the lattice of my fingers, where's my eyeball there? These are muscles that in one direction they will shorten and in another direction they will bulge. And that kind of antagonistic or opposite pattern of shortening and bulging it and squirting it forward can lead to like an inchworm kind of emotion. And so we see these rings, and because it's a triploblast, so there is a coelom, a body cavity filled with fluid. So you can think about that analogy of the water balloon, and your kind of fingers being the muscles on the outside, either squeezing it forward or shortening it, if you could do that with a water balloon. Okay, so there's the circular, there's the longitudinal, and there's that coelom that it's firing across. Here's the corona. So this is the wheel organs up here, and you can see leading into a center like kind of a Return of the Jedi, kind of a mouth. And then we've got a mastix and a gastric gland. Both of those are associated. The mastix is kind of like a grinding area to break down the tiny little protozoans or small things that it might be eating. And then the gastric gland facilitates the initiation of the digestion through the rest of the alimentary canal, which has a front end and a back end. The syncytial epidermis can secrete a cuticle, and so we know something about the distribution of rotifers, not just from catching them and watching them in a live state, but looking at these keratin-based lamina larissa. So these kind of the ornateness, how ornate, how horned or sculpted the cuticle is, and we can trace their abundance in lake sediments as well. So blunt spines or quite extravagant spines like that. So a blunt spine, so this would be a lamina and this would be a larissa. From a reproductive perspective, the rotifers are kind of amazing. So they are gonochoric. The opposite of, remember, with the platyhelminth, we talked a lot about hermaphrodism, and we will continue because hermaphrodism is certainly not restricted even in the bilateria. It appears again and again and again as a strategy. So they are gonochoric, but we don't always see males. We see that the role of the male is hyper reduced for many rotifer lineages. Where the males do operate, we see, as we did with some of the free-living platyhelminth, the hypodermic impregnation. So not an insertion, not kind of opposite genitalia that meet and exchange gametes, but just breaking into the body cavity and injecting sperm into the coelom. Now, one of the other amazing kind of things that we're somewhat familiar with because of dealing with some protozoan lineages and some parasitic lineages is this alteration of sexual and asexual generations. So sex, not all of the time, right? Sex when it's needed. When might sex be needed? You can think about environmental conditions, the scrambling, that meiosis being kind of a helpful resetting of your Scrabble board to give you some new words to deal with some new environmental challenges. And what you're looking at here, these air balloons are examinations of a dissected set of reproductive organs out of a female and a male example of the rotifer. I love this quote. This is, again, from Rupert Fox and Barnes. They had some bangers. They had some good stuff. Males, if present at all, are dwarf, aberrant, short-lived, and little more than a swimming testes and oversized penis. Not so useful, right? Except with the idea that perhaps sex provides this rescrambling, the resetting of your Scrabble board to deal with a new set of environmental conditions. So the bedolid, yeah, exactly. So kind of like anglerfish. I just glanced down to see that comment. So kind of like anglerfish males. That's right. So not always sex, but sex when necessary. So one of the cool things about, and we're not going to go into and perhaps you covered it a little bit in evolution. Tell me in the chat if you did. But there are genetic ways for estimating for certain lineages or species how frequently sex has occurred in evolutionary time. And so there are lineages of rotifers called dolid rotifers or bedolid rotifers that looks like there is a genetic or genomic incompatibility that will interfere with meiosis, which is this precursor to sexual reproduction. And the bedolid rotifers are sometimes lineages that we think sex has occurred in the past, but not for hundreds of thousands of generations, like many, many, many years. Now, part of that, what does asexual reproduction provide? Well, if you're good at, if you're well matched with your environment, you can expand to fill it very quickly. You don't have to waste time with finding, in a gonochoric species, finding another member of the opposite sex and doing whatever it is necessary to kind of share your gametes. You're just like, today, me and more. The amazing thing, or amongst the many amazing things about the rotifers, is some of the traits that they have evolved associated with dealing with periods of incompatible environments. Sometimes that's sex, but sometimes that's hunkering down and becoming dormant. And rotifers have this incredible capacity. We first learned to deal with ionizing radiation. So they can deal with doses of ionizing radiation that is the first line of the bullet there would result in like hundreds of strand breaks in a genome with only, in us, for instance, but for the rotifer, that would result in only a 20% reduction in fertility. And since that discovery, what we've uncovered, and the ionizing radiation is talked about there in that PNAS paper on the left, this seems to be taking a tool from your toolkit that is good for one thing and saying, oh, it's also good for this other thing. So the other thing is the ionizing radiation, but it is the primary adaptation seems to be surviving episodes of desiccation with the lack of water. So characteristically in these habitats, remember where they live, marine, freshwater, semi-terrestrial, meaning of course kind of semi-aquatic. So lack of water can be quite stressful. And the way that they deal with that is a DNA repair mechanism and series of enzymes that is also very good at repairing damage from ionizing radiation. There's Caramel to tell me to take a breath and to take a sip and to look down and move on to thinking more about motion. So the motion of the rotifer, and this also gives us a chance to look at not just the still images. So I hope this is refreshing well on your screen. Let's talk about the feeding currents here. And we'll zoom in on the left-hand video. And what you're looking at there is the corona, is that ciliary organ. And you can see with the algal cells that are out here moving in the current that is set up by the beating of the cilia and the beating of those rotary organs. And you can see the mouth kind of working away, and then the gut kind of churning and churning and churning, and then some bit of the rest of the body inching in and out. Q, Smith scrolling out and across. So let's take a look at telescoping. Now remember we talked about the antagonistic firing of those muscles. Let's go and start that one. Start. Okay, so there, oh, look at that. So that was longitudinal muscles, right? Shrinking away. But then it inches out, and that kind of like inchworming, being able to move and pull in and extract out or extend out, that's the antagonistic firing of just really two principal groups of muscles, the circular and the longitudinal. And I think that's me. And this speaks to, as we're looking, some of these are suspension feeders. So you've got the ciliary currents that are set up. So this, you could imagine, on the left-hand side is kind of the suspension feeder, right? But they can move around and they can be raptivorous. They can actually even swim somewhat through the water. So you can set up or imagine quite an ecological network of different species of rotifers that are playing different roles. Some are suspension feeders, feeding on very, very small things. Some are raptors, kind of moving around and preying on bigger things. And some are omnivores, do a little bit of both. So not to put too fine a point on it, but here is an older drawing that the last was the cross-section, the kind of crokinole disc of your rotifer. But you can see here in this kind of Arnold Schwarzenegger examination of the bands of muscles. So you can see their kind of extension, like inner tubes around the circular muscles, and that long lengthening that even can be used to retract the trocha, or the face, the coronal organ. So that coelom, the pseudocoel that it talks about there, is this hydrostatic skeleton. We saw the hydrostatic skeleton last week with the platyhelminth flatworms. We're going to see it again and again. This is a really important way of moving through the world when you don't have a hardened carapace or an internal set of bones. And in this course, if you've got an internal set of bones, you're out. Last view of this is this kind of diagram. So this isn't an inching rotifer, but this is an inching worm, where because it's segmented, and remember the rotifers are not segmented, you can kind of see I use the segmentation here just to show you both in the video and in the drawing the change in the body position associated with these waves of muscular contraction. So waves kind of proceeding along the length of the body, whereas one part of the body is squeezing together, another part of the body is kind of pushing or inching back, condensing itself and then being pushed forward. So this inchworm kind of motion is a really successful way for moving when you don't have a carapace and when what you've got is a hydrostatic skeleton. Let's talk about durability again in a couple of different ways into the last kind of bits of thinking about the rotifers. They're very good at dealing not just with ionizing radiation and not just with desiccation, but with freezing and likely similar kind of genomic repair mechanisms. So this is a story from Current Biology, I believe, of a couple of years ago that talked about the reanimation, the waking up, like talk about a snooze, of a rotifer from 24,000-year-old collections of Arctic permafrost. So in Siberia, this rotifer was recovered in an ice core. The ice core was carbon dated to more or less 24,000 years ago. They brought it to room temperature, and it begins to move around. And this is at the time, this is the record. I'm pretty sure it's still the longest record for rotiver survival in a frozen state. And this bouncing, beautiful baby rotifer, this is it. This is you watching a video in 2026 now of a rotifer that the last time it was alive was 24,000 years ago. Like, talk about an escape mechanism, right? That's so not physical escape, temporal escape. That's amazing. So this is kind of looking at, that's the actual rebirth. So that's it in its very reduced, desiccated state that's dealing with freezing. Water is added, and then the celebratory dance after 24,000 years of hanging out begins. I like that. I like the joke in the chat. Yeah, just 5 more seconds. No, 5 more 1,000 years. That's a hell of a snooze. Now, also, rotifers Dormancy, why? Okay, well, dealing with desiccation, avoiding desiccation. If you live in ephemeral habitats that dry up, dealing with, or that freeze periodically because of where you live latitudinally, dealing with those problems by escaping and becoming dormant is probably pretty powerful. In a really cool way, one of the new things that we've discovered in the last couple of years is aligning with Pedro Pascal and et al. In The Last of Us. Rotifers are prone, some of them prone to infection with a really aggressive fungus. And the longer that they go dormant, the more likely they are to avoid infection. So dormancy may not just be a useful adaptation towards dealing with desiccation and freezing, but also with infection. With fungal infection. The mechanism for this, not entirely understood yet. So if you're liking the rotifers and you're liking the kind of rotifers, they do it all, then perhaps this is something that you can go and explore. Last bit for the rotifers is the idea, and it's another cultural reference, is this hypothesis that dates back for nearly 100 years called the everything is everywhere hypothesis that I'm going to adapt so that we do justice to Everything Everywhere is All at Once and Ke-Huy Quan and Michelle Yeoh's incredible movie of a couple of years ago. So not just the everything is everywhere all at once hypothesis. And this is a classical view that organisms that, say, less than 2 millimeters long often have a cosmopolitan distribution, particularly when we look at them using classical taxonomy. Now put a pin in that and let's come back and examine that assumption in a second. But the mechanism behind this everything is everywhere all at once hypothesis is that because of very small size and often the ability to enter a dormant state, these very small organisms, these propagules, can be moved around. And historically, rotifers have been used as an exemplar taxon that potentially supports the everything is everywhere all at once hypothesis. Now, I'm going to try something here. I'm going to draw out. We've been talking about a lot of these things from a phylogenetic perspective. And so what we've got here is a small phylogeny of A, B, C, D, E, F, right? Peekaboo. Now, what would, if I put a matrix of places on the outside, 1, 2, 3, 4 different places here on the outside. Come on, pen, don't fail me now. Okay, so different species, different places that the species live. What would phylogenetic support for the everything is everywhere all at once hypothesis look like? Would you be filling in cells longitudinally, like along the column, or would you be filling in cells across the column? Tell me in the text. The text, what are we talking about? Tell me in the chat. Yeah, don't text me. Put it in the chat. So what's the support for the everything is everywhere all at once hypothesis for multiple species across, say, continents? Would the support be that they occur along the columns or across the columns? Oh, thank you. So you can see it. All right. I'll keep that around. And let me go and examine some of your hypotheses. Yeah. It's certainly, if these are different continents, support for everything is everywhere all at once would be one of these species where we found kind of filled in bars all the way across. It could also be, yeah, and that's really it. It's a cross. A cross is the key mechanism or the key observation that provides support for the everything is everywhere all at once hypothesis. Cool. Nice. Thank you. All right. The next bit of business is for me to check the chat. I don't think I missed anything in there, but if I did, please let me know. is to switch gears slightly and talk about the Ketognaths. So remember, we were down here talking about the rotifers that can live practically everywhere, relatively diverse, and more are coming. And then Ketognaths, they live marine, not a whole bunch of species, but they've been around a long time. And they have been a bit of an evolutionary mystery. This is an enigmatic protosome. Now, who's the quotes there? That's not a Joey Tribbiani quote. This is a quote from Casey Dunn, the lead author of the Dunn et al. Phylogeny, expounding back in 2014 about the position held by the Ketognaths in the phylogeny of animals has consequences for our understanding of jaws, like the gnathos of jaws. So let's first of all introduce ourselves to the beautiful things. And as I'm doing this, could you throw me a yes or a no in the chat? Have you heard of Ketognaths before, or arrowworms, as they're sometimes called? Yes or a no? One of the cool things about these, so they're very, very small, but we're looking at, they have a little kind of a fin that moves them forward. They are predators, and they have these grasping spines, gorgeous grasping spines up at the front end. This is a male, and this is a female. They have no discrete, they're small enough, so no discrete gas exchange or excretory system. They can be hermaphroditic. They have this central nervous, like a relatively complicated central nervous system. These are predators, mobile predators. Awesome. So I see a mix of things. Lots. Some have heard of arrowworms. Some have heard of Ketognaths. Some have heard of nothing at all. Well, for everyone, welcome to the Ketognath zone. Pigment cup ocelli. I'm going to show you my simplified version here in a second, but these are some kind of, so pigment cup ocelli, that's a light, that's a photoreceptor. Not a complicated image forming thing, but responding to shadows and directionality that light would provide in a marine environment is a key thing for the Ketognaths. So corona ciliata and these grasping spines, lateral fins helping them move. These are chitinous things. They can capture prey and they can envenomate them. So that's associated, like adaptations associated with the spines. These ciliary fences that are up at the top, they are associated with sensing prey. They're a sit and wait predator. So they need a trigger, something to tell them to jump forward and to begin to eat. Okay, so little tiny torpedoes, essentially. Let's take a look. This is actually, these are SEMs not of extant or living Ketognaths, but of fossil Ketognaths. Ket for long hair, Gnath for jaw, so this is hairy jaws. They're found in all marine environments. They're major predators, and we have this good fossil record, like these individuals down here, since the Burgess Shale. And we have moved them around. They have a tortuous taxonomic history where they have been moved between multiple different phyla. Again, this is a common thing. Remember the story with the Xenoturbella, because the first phylogenetically, molecularly informed phylogenetic analyses included contaminant sequences, prey items. Just like the Xenoturbella we thought was a mollusk, not because it's a mollusk, but because our evidence we didn't know was derived from the stomach. And Xenoturbella eats mollusks. Now, what eats Ketognaths? What do Ketognaths eat? They are voracious predators of the holoplankton. So that part of the pelagic zone, they're always out there in the pelagic zone. And they prey on a diverse set of things. So the arrows, the directionality here in the color says Ketognaths prey on copepods, amphipods, gastropods, so the arrows point towards who's eating whom. But you can see many other things prey directly on Ketognaths or prey on species that feed on Ketognaths. So sea lions don't eat Ketognaths, but they eat the fish that eat the Ketognaths. So there's multiple specific interactions across trophic zones. And they can have specific parasitic interactions as well. That there are multiple lineages of parasite that parasitize Ketognaths and the food that they are eating. So these are some SEM's beautiful things of the grasping spines. Those are the triggers, right? And I said it was a sit and wait predator. Let's see the little kind of O on the grid here. It's going to keep looping through. This little torpedo, bam, eats up the copepod. And the bottom right-hand side, this video, is showing those grasping spines, and I've slowed that video down under a light microscope just to show you the manipulation into the face, into the mouth that feeding does. So very small organisms, millimeters long. But with a long fossil record, and some of the fossil ketognaths were not small. They were 30 centimeters long. So if we've got modern animals that are millimeters long, like 2 centimeters, these were orders of magnitude bigger, possibly fairly terrifying to be swimming around with, particularly if you were smaller than 30 centimeters long. We think of them principally up in the upper levels of the plankton because that's where we see them. That's a bias of where we examine. And this is a video from nearly 10 years ago now, where one of these streaming high video remotely operated vehicles that at this point was nearly 6 kilometers beneath the surface found this ketognath, an arrowworm that is clearly using other methods of responding to its prey than those photoreceptor cells, because we are kilometers underwater here. There is no light. So it's poised here. This is kind of sitting and waiting for something to brush up against that ciliary spines so that it kind of jumps forward to grab it. This is probably the deepest that an arrowworm has ever been found. Now, from a phylogenetic perspective, there seems to be a lot of traits here, right? Like, this is not the Placozoan where we need to figure out how many species there are, but we look at them and there's like only 6 cell types. So how do we, like a really reduced thing? These look relatively ornate, like little tiny torpedo fish, right? Not a fish, not a torpedo either, but lots of different forms and functions here. And so what I'm showing you here across this phylogeny of different genera of ketognaths is the gain of a trait, the retention of a trait, the plus, or the loss of a trait. And so one of the cool things about the real pattern here is just focusing in on the proportion, the number of times where ketognath evolution seems to have moved forward, not by the addition of a new trait, but by the loss of something that its ancestor species, its ancestor taxa had. And so you don't have to worry about the names here at all. This is me just pulling on another thread of commonality throughout the course, reinforcing that evolution is by loss nearly as frequently as it is by gain. So ketognaths are protostomes, but are they our early branching lineage? This is our simplified diagram, right? So we've got protostomes, this as compared to deuterostomes, so we're in the bilateria. But what do we do with ketognaths? They're sitting out here. So where do they fall? Are they ecdysoans? Are they spiralians? Are they gnathiferans? What do we do? This is Casey Dunn, and this is what he wrote in that paper associated with your phylogeny, our map in this course, is that interpreting them as a deep branching, an early branching lineage is consistent with a bunch of things. Their appearance of spines back into the Burgess Shale at the Cambrian explosion, as well as some more or less deuterostome-like traits featured in their development, like deuterostomy, enterocilian radial cleavage. We haven't pulled on those traits yet. Historically, developmental traits like that were a major division in between the deuterostomes and the protostomes. What we do know, and we've talked about it very briefly together, is that things are fuzzier than we used to think 50, 100 years ago regarding these developmental traits. Here are some of the traits as kind of as bars or presents across. So here's where ketognaths sit in that Dunn phylogeny of 2014, outside as a protostome, but we don't know where they sit in relation to the spiralians. But they have basement membranes and nerves and brains. They lack all of these through gut, a mesoderm, a coelom, and a blood vascular system. Now, in 2017, there was some new exciting research that was produced using Hox genes. Now, you remember Hox genes, you've used them before, you've been exposed to them before, but just as a reminder, they're conserved regions, regulating gene regions that control some of the most fundamental parts of subsequent embryonic development. Consequently, we share Hox genes with a wide variety of taxa. These are old bits of machinery that we share because of the unity of life. And so comparing Hox genes patterns is a really effective way to infer deep, old phylogenetic relationships. Now, what was known when Andreas and Peter here did produced the results of their studies, they were focusing largely on rotifer Hox genes, so Gnathiferan Hox genes. And what they found was here. So I'm going to ask you this question here in a second. So we've got genes, different Hox genes covered as different boxes. And we've got members of the ecdysozoans, spiralians, gnathiferans, and then we've got some deuterostomes and some xenocoelomorph and cnidarians out here. This box right here, medpost, that's a gene, that's a Hox gene that was historically known only from rotifers. So even though they're very conserved, there are lineages that have, old lineages that have their own. What we saw here, take a look. So we've got rotifer, ketognath. We've got 1, 2, 3, 4. All of those, the rotifers and the ketognaths, all have medpost, the historically only rotifer Hox gene. So, I'm apologizing. I'm flipping that story. And thank you past me for putting the bullet in so that I can remind that. This was medpost was a ketognath Hox gene that was then all of a sudden appearing in the rotifers. Now that has been interpreted as being evidence that along with the jaws, remember we talked about the importance of jaws, suggests that these are Gnathiferans, the rotifers should and others should also include the ketognaths. So it suggests that we've found a home for ketognaths as a member of the Gnathiferans. So a sister group to the remaining spiralians. So we've got, this was what we started out, ketognaths, where do we live? And then we know ketognaths, in the simplified view, ketognaths are here in the spiralia. And so here we saw the ketognaths. Now we used to see the ketognaths kind of outside as the early, like an unknown, maybe an early branching protostome lineage. Now it suggests that we should consider the ketognaths because of Hox genes and jaw apparatus as part of the Gnathiferans. So that's thinking about the Gnathiferans, two different phyla, formerly disparate. When I took this course, back in the 20th century when I would have taken this course, it would have been weeks in between when we talked about these things. And now we know, with new tools helping us evaluate old and new evidence, that these are close relatives, not distant relatives. And let's take and set the stage for Wednesday by considering now the Lophophore and the taxa, the phyla, the species that have a Lophophore. First of all, what is a Lophophore? A Lophophore, I'm going to, in the next 5 to 10 minutes, talk about that. 5 to 7 minutes even. Imagine what you're looking at there. So we've got beating cilia. If you zoom in here, see the cilia, they're kind of moving around. So these tentacles surrounding a mouth. So they have cilia are moving around like a little manufacturing line. That manufacturing line is moving little particles into the mouth. We've seen this kind of thing before. This kind of tentacle with a mouth at the center. It's a great way of being a sessile organism. But remember, we're in the bilateria here. So tentacles radiating out from around a mouth, that's not usually a signature of a bilaterian organism. Now that we'll unpack on Wednesday. I know that's some foreshadowing there. That's a teaser. But just to think about the organ, the Lophophore is also unique in that there's part of the body coelom in each one of these tentacles. So the tentacles are coelom filled around a mouth with cilia that beat, that bring it in water, that create a water kind of a current, capture these things and bring them into the mouth. And so here is looked at in as a line drawing. So we've got the water shown in these arrows here. That's the movement of the water kind of being brought into the tentacle. And then if we take a cross section of tentacle from right here and move it up here, you can see that center, this little Cheerio, kind of a furry Cheerio. Don't eat the furry Cheerios. This cross section, this is part of that hydrostat, this body cavity, this coelom, this fluid-filled body cavity that's inside of the coelom. So we've got that, and then we've got a mouth and kind of a U-shaped, and for the taxa we'll talk about on Wednesday, the gut is U-shaped. So you see the anus isn't kind of way over there at the end of the body, but the gut bends around, is U-shaped and comes up right here. And they're gorgeous. This particular view of a Lophophore is a bryozoan. You can follow the video and watch that later on, but it's just, I could watch Lophophores for a while. So Lophophorata, in quotes, would be one name to potentially describe a monophyletic group for taxa that had this character, that had a Lophophore. Cilia, beating cilia, circularly arranged around a mouth where the coelom extended into branches of the tentacles. So this crown of ciliated hollow tentacles that does food collection and gas exchange, that tend to be sessile, tend to be sedentary suspension feeders, no distinct head. They have nothing to worry about in the zombie apocalypse. Any of the Lophophore-bearing taxa, they don't have a brain. And they use the cilia to capture phytoplankton and other small planktonic organisms. So we've got these tentacles. And one of the key questions in animal phylogenetics within the Spiralia is regarding the relationships for the animals that have this feature. Because there's two principal hypotheses. One of them on the left here is that we see a group called Trochozoa that has annelids, mollusks, nemerteans, brachiopods, phoronids. And then we've got a big polytomy of things that we don't understand where they particularly sit, including the bryozoans. Now, you don't see the word Lophophorata here on what I'm showing you right now, because the taxa that have the Lophophore are the phoronids, the brachiopods, and the bryozoans. That's not a monophyletic group. This is the other contemporary hypothesis, is that within the Spiralia, we do have uncertainty about who's related to whom with some of the taxa, but there's two lineages that are supported. The Trochozoa, so the annelids, the nemerteans, and the mollusks, very diverse group that we'll get to in and around the reading break, and the bryozoans, the brachiopods, and the phoronids, the Lophophore-bearing taxa. Oops. So depending on the placement, and you see which one. So in each, the brachiopods and the phoronids, they're together. Like that evidence is pretty resilient. It's really the bryozoans, these moss animals, that we're uncertain about where they sit. So depending on the placement within the Spiralia, we can either talk about Lophophorata or Trochozoa. And that's all based on what happens to the bryozoans. So if we map the Lophophore like this, you can ask the question, I will ask the question of you to consider and to mull over as we enter into Wednesday's shared time together, is which one of these appears in the Dunn phylogeny, and is the one that's in the Dunn phylogeny, is it the most parsimonious? I'm not going to answer that. I want you to answer that and think about it while we're talking together on Wednesday, because we've used up most of our time. Caramel's exhausted. And so we've spent some time, we cleaned up some amazing colonial Serracicari larvae from the trematodes as an outstanding little story. Then we talked about the Gnathiferans, the rotifers, and different ways that they have for moving, feeding, and dealing with harsh conditions. We talked about ketognaths and Hox genes and the jaw morphology as being two genomic or genetic and morphological features that unify them now, not as being less enigmatic and as being part of the Gnathiferans. And then we gave a brief introduction to this hollow-filled tentacle head of the Lophophore. And that's what we're going to unpack next time by talking about the brachiopods, the bryozoans, and these muppets, these little phoronid worms. So what else to think about? The case study's open until tomorrow. Think about your species assignment. Don't forget it. It's still coming in March, but now I'm told it's February. So it's like we're 1 month closer to when it's due, because in January it was due in March, and that's a long way away. Now we're February. It's less far away. And prepare for your labs this week, and greet all with enthusiasm all of the new lab team. Do your watching of the videos beforehand, particularly the Ripley's video and the kind of the notes to make about that. If you're going to participate in the optional, the scavenger hunt and the models, you can do those. And that's really it. That's the time we've got together. I'm going to stop recording in a second, and then I'll hang out if there's any questions. So until we see each other again, take care of yourself, take care of others around you, and think invertebrate thoughts.